Good day, everyone. I'm here today to discuss to you the non-Mendelian patterns of inheritance. Now, there are different non-Mendelian patterns, which is the principle of incomplete dominance, principle of codominance, principle of multiple alleles, sex-linked traits, sex-influenced trait, and sex-limited traits. Let's first discuss the principle of incomplete dominance. One pattern discovered is that one allele is not completely dominant over the other allele, resulting in the blending of characteristics or the appearance of an intermediate phenotype possessed by two parents. So this means that if two alleles are being crossed, none of them would be completely dominant over the other allele, for both of them would blend. Now, I'll give you an example wherein we're gonna cross a red flower to a white flower. Now, we'll just simply use a Punnett square wherein we just cross R with W, resulting to this, and same goes to the other side. Now, we've got this outcome of offsprings, a cross between a red colored flower and another white colored flower would result in the production of offsprings that are pink colored. Now, the offsprings are all pink colored flowers, a heterozygous characteristic which is distinct from the two homozygous parents. When the offspring were allowed to self-fertilize, the offspring produced a genotype and phenotype ratio of 25% red, 50% pink, and 25% white. So next, we'll move on to the principle of codominance. So, in some cases, the heterozygous characteristics are fully expressed in the offspring in a phenomenon called condominance, as in coat color in cattle. So, this is where both alleles will be seen and would not blend. Now another example, the same example, I'm just gonna change the red flower into a red coat and white flower into a white coat. Now same procedure, we'll just cross and would get the outcome of this. Now the phenotype will be a 100% roan coat. A roan coat color is one in which the coat color is the mixture of the red and white color. So the cow would have a cattle of the offspring cattle of the red coated and white coated cow would have, or the red and white coat would be seen both on the cattle. Now, let's move on to the principle of multiple alleles. The traits that have been discussed in the preceding sections are traits that are controlled by two alleles for a given trait. Through the help of modern technology, genetics discovered that there are some traits that are not controlled by two alleles, but by multiple alleles. So, one example of traits controlled by multiple alleles is the human blood group. So we're gonna have a sample cross between a heterozygous blood type A female and a heterozygous blood type B male. Same thing, we're just gonna cross blood type A to blood type B, and then everything, blood type A, blood type B to I. And now we've got this outcome of the multiple allele cross for ABO blood system. We have a 1 blood type AB, 1 blood type A, 1 blood type B, and 1 blood type O. So the phenotype of the 4 of them would be 25% each. So next we'll be discussing sex-linked traits. Sex-linked traits are traits that are controlled by the genes found in the sex chromosomes, both X and Y. Sex-linked traits are recessive in most cases. The recessive gene or allele was inherited from one or both of parents. Sex-linked traits affects mostly male offsprings. Example of sex-linked traits is a disorder which is hemophilia. Hemophilia is a recessive genetic disorder wherein the blood does not clot. This is caused by the lack of genes that synthesize the protein that is needed to initiate the blood clotting process. Now, if you want to learn more about this hemophilia, you can watch the royal diseases from tainted blood in YouTube. Now, we're going to have another example on how we're gonna know if a person would be a carrier or would be hemophiliac. Now, all female offsprings are normal, 50%. Although one is a carrier, one male offspring is normal, 25%, and one male offspring at 25% is hemophiliac. Thus, the probability of having sons who are hemophiliac or bleeders is 25. Now, so we got this outcome. So 3 of them would be normal, and this one is the carrier of the hemophiliac, or he is the hemophiliac person. Now, let's move on to sex influence trait. Sex influence trait is carried by the autosomes and not the sex chromosomes. So we have this table of female and male. These are genetic table for baldness in male and female humans. Now, if you're a female and you have a genotype of X small letter b and X small letter b, you would be bald. If you have the genotype of X capital B and X small letter b, you would not be bald. And if you have a genotype of X capital B and X capital B, you will still not be bald. But while you're male, if you have a genotype of X capital B and Y small letter b, you will be bald. if you have X small letter B and Y small letter b, you would be bald. But if you have X capital B and Y capital B, you will not be bald. So we'll have a sex influence cross for baldness. So same, we'll just simply cross. Result to a not bald, 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 and bald. As you can see in the table. Now here we have 2 male bald, 1 female bald, and 1 female not bald. So lastly, we'll move on to sex limited trait. Sex limited traits are determined by the genes in the autosomes and therefore are found in both male and female. Phenotype of the genes in the organism. The trait is expressed only in one gender and does not appear in the other. The genes may have dominant effect in one sex and recessive effect on the other. Only typical example is the production of milk lactating mothers, but the effect of the genes is only observable among females since females exhibit or possess the normal conditions for such effect to happen, which is the presence of well-developed breast. So that is all, and I would like to thank you for joining us today, and I hope you learned something, and I ask you please to like and subscribe this video. Thank you.